Good day everyone. So we will now be reporting about the family Orthomyxoviridae and Pneumoviridae. To start, characteristics of the family Orthomyxoviridae. So Orthomyxoviridae is derived from the Greek orthos, meaning correct or right, and myxa, meaning mucus. They are also linear, negative sense, single-stranded RNA, and they have a segmented genome arrangement. When we say segmented, yung genome or genetic material ng virus ay non-continuous, or it is composed or divided into fragments that make a complete viral genome. Then the size of orthomyxoviruses range from 80 to 120 nanometers in diameter, and they are enveloped and meron silang helical symmetry. They also replicate in the nucleus of the host cell. Since yung mga orthomyxoviruses, they cannot make their own mRNAs or messenger RNAs. Lastly, orthomyxoviruses contain significant pathogens of both humans and animals. So, family Orthomyxoviridae is composed of 6 genera. Influenza virus A, common pathogens of horses, swine, humans, and domestic poultry worldwide, and nagkakos din sila ng sporadic or geographically limited infections and disease sa mga mink, seals, whales, and dogs. The next genus, Influenza virus B, pathogens of humans, but currently may mga reports na ng influenza B virus sa mga seals. The next, Influenza virus C that infect humans and swine, pero mostly, mild lang yung infection with this genus. Then, sa mga influenza virus na to, influenza, meaning influence, na nag-o-originate from a worldwide pandemic noong 1580, na in that time was believed na yung disease was an influence of the stars, and hindi dahil sa mga microscopic pathogens na nag-cause dito, kaya influenza. The next genus, Togotovirus, tick-borne viruses that sporadically infect livestock and humans, mainly sa Africa, Europe, Asia, and most recently sa North America po. Then, fifth, Quaranja virus, a recently established genus, includes viruses predominantly isolated from ticks and birds, pero there are also reported cases na na-isolate din siya sa human with febrile disease, or ito yung nag-show ng symptoms na fever. Then lastly, is a virus genus with the sole member, which is infectious salmon anemia virus, a highly fatal disease ng mga marine farmed Atlantic salmon. And meron ding proposed new genus na Influenza virus D, newly discovered pa lang siya, isolated from cows and bats, and wala pa siyang definitive or exact taxonomic classification. Then, next slide. A classification system was developed for influenza viruses. In the current classification system, influenza A viruses are categorized into 16 hemagglutinin and 9 neuraminidase subtypes. So, ito yung mga surface glycoproteins na nasa genus influenza virus A and B mainly. Hemagglutinin or HA that has 16 antigens, age 1 to age 16. Yung main role niya is that it binds to the host receptor, initiating infection following the attachment of the virus to the susceptible cell. The neuraminidase or NA that has 9 antigens, N1, 2, and 9. It promotes penetration of the virus and release of replicates from the infected host cell. So, mahalaga itong mga to sa mismong viral replication ng influenza A virus. And naka-classify yung mga influenza A viruses into subtypes based sa mga combinations ng mga proteins na nasa surface ng virus. Yun nga nasabi ko na hemagglutinin and neuraminidase. So, itong slide na to, ito yung mga example ng mga important influenza virus A subtypes na mas madidiscuss pa mamaya isa-isa. Then next, capsid and genome. Orthomyxoviruses are pleomorphic. Filamentous sila pero nagiging spherical when cultivated sa laboratory or laboratory cultivation. Then size, 80 to 120 nanometers in diameter, and they are linear, negative sense, single-stranded RNA na nasabi na rin kanina. They consist of an envelope with large spikes surrounding 6 to 8 helically symmetrical nucleocapsid segments of different sizes. So, ulit, segmented ang mga orthomyxoviruses, like yung genera Influenza virus A, Influenza virus B, and Isa virus, may 8 segments sa viral genome nila na segmented. Then, 2 kinds of spikes, rod shape, yun nga hemagglutinin glycoprotein, and mushroom shape naman yung neuraminidase protein. So, for the replication, orthomyxoviruses replicate in the nucleus of the host cell, since again, they cannot make their own mRNAs. So, they use cellular RNAs as primers for initiating the viral mRNA synthesis in a process known as cap-snatching. Yan. For attachment and entry, nagbabind yung hemagglutinin glycoprotein or spike ng influenza virion sa receptors ng host, specifically sialic acid-containing receptors ng host. Then, human-adapted hemagglutinin or H1 and H3 viruses show binding preference sa long oligosaccharides present on epithelial cells in the host upper respiratory tract or URT. Then, influenza viruses enter cells via receptor-mediated endocytosis, gaya din ng ibang mga viruses, since common nga ito. Then, with all the viruses, the genome of orthomyxoviruses, having a negative sense RNA genome, it serves 2 functions. As a template for the synthesis of messenger RNAs, mRNAs, and as a template for the synthesis of positive sense naman, intermediate RNA, which is the template for progeny genomic RNA synthesis. Then, orthomyxoviruses replication takes about 6 hours and it kills the host cell. So, ayan yung illustration ng complete cycle ng mga orthomyxoviruses. Then, before we proceed sa mga diseases, as a review lang, na-discuss sa microbio yung term na antigenic drift, wherein yung mga organisms, during replication, nag-undergo sila ng mga frequent small mutations or minor changes, like yung point mutation, example, wherein yung AT is substituted ng CG, so mali yung magiging result na amino acid into protein. Then yung frame shift mutation na may na-insert or na-delete na maling nucleotide, so mali rin yung protein na napoproduce, leading to antigenic or genetic variation. So ito yung antigenic drift that is gradual, continuous, and occurring frequently na minor antigenic change lang ang nagiging result compared sa antigenic shift na sudden, discontinuous, and occurring less frequently, leading to major antigenic change. And again, nangyayari yung antigenic drift dahil sa mga mutations, nangyayari yung antigenic shift because of gene reassortment. So, gene reassortment ay, di ba, as for example, dalawang magkaibang influenza virus yung nag-infect sa isang cell. Then, doon sa time na yon, doon nagkakaroon ng opportunity na mag-shuffle or mag-switch yung genes nila, since nasabi ko na nga na segmented yung genome ng mga orthomyxoviruses, particularly influenza A virus. 8 yung segments ng genome niya, so high chance na magkaroon ng gene reassortment with other virus, leading to a novel or new virus. Kaya ang dami ring subtypes under this genus. Well, sa antigenic drift, it just leads to a new strain with the same subtype, since small changes or mutations lang ang laging nangyayari. Then, antigenic drift occurs in both influenza A and B, while antigenic shift, kay influenza A lang siya nangyayari. This is also why influenza A viruses can cause major epidemics and pandemics. Kaya ang dami nang nangyayaring mga outbreaks or influenza pandemic. Parang lagi silang nakakagawa ng novel or new virus because of genetic shift na kapag mag-i-infect sa tao yung viruses, hindi nare-recognize ng body since novel nga siya or new subtype, ganon. So, mahina pa yung magiging immunity ng mga tao sa virus na mga to. Now, moving on with the members of the genus Influenza virus A, we have equine, swine, avian, canine, bat, and bovine influenza D virus, then lastly, human influenza virus. So, equine influenza viruses are common cause of acute respiratory disease in horses and other equids, with influenza being the most important cause of viral respiratory disease in horses. Equine influenza viruses spread very rapidly and cause infections with high morbidity rate, 24 to 48 hours after infection, and rare or low mortality rate naman, but prolonged fever in pregnant mares may actually result in abortion. Then age 7 and 7 subtype of influenza A virus or the equine influenza virus 1, siya rin yung cause ng outbreak in 1979 sa Italy, na-isolate or nakita sa India noong 1987 and Egypt last 1989. But this subtype no longer circulates in equine populations or extinct na siya and wala nang reported cases of infection sa equine sa subtype na ito. Then next, age 3 and 8 subtype or the equine influenza virus 2. Next, equine influenza viruses are highly contagious. They spread rapidly by infectious exudate that is aerosolized by frequent coughing. And the virus is excreted during the incubation period na as short as 24 hours up to 3 days. Horses remain infectious for at least 5 days after clinical disease begins, and close contact between horses facilitates rapid transmission. Then, fomites, such as contaminated clothing of personnel, equipment, and transport vehicles, may also contribute to virus dissemination. The next, for susceptible horses. Horses age 1 to 5 years old, lalo na kapag nakahouse sila in large groups, and at risk din yung mga equine populations that are moved frequently, such as race horses, breeding stock, show jumpers, and horses that are sent to sales. Then also, nagiging immunosuppressed kasi yung mga horse during traveling or training nila na pwedeng mag-cause ng increased risk of infection or mas maging severe yung effect ng virus or infection. Then next slide, for the pathogenesis and pathology. The virus mainly replicates in epithelial cells of the upper and lower respiratory tract of susceptible horses. Then, meron ding mga horses na asymptomatic, immune sila and hindi magkakasakit, pero pwede pa rin makapag-replicate yung virus sa kanila and then mag-shed na pwedeng maging source of infection ng ibang mga horses. So, there are factors actually that contribute to the innate resistance of horses, such as the mucous blanket that protects the respiratory epithelium and the continuous beating of cilia that clears virus from the respiratory tract of horses. Then the soluble lectins, a class of protein na yun nga has a role for the innate immune responses against pathogens, lung surfactants, complex substances na naturally sinisecrete ng lungs, and shallow glycoproteins. Also, alveolar macrophages that are actually the most abundant innate immune cells in the lungs, and secretory immunoglobulin A is proposed to be the most relevant antibody in upper respiratory tract. Then next, laboratory diagnosis. Yung clinical diagnosis sa mga acute cases, mabilas lang siya. Pero sa mga horses na may influenza virus, pero mild lang yung sinushow na symptoms, mas mahirap yung diagnosis since it has to be differentiated with the other respiratory infections, including those caused by equine herpes virus, equine rhinitis viruses, and iba pang mga respiratory disease-causing bacteria. The nasal or oropharyngeal swabs usually pinagkukuha ng sample, and specimen should be placed in a virus transport medium. For equine influenza viruses 2 or age 3 and 8 viruses, they can be grown or replicate in 10-day-old embryonated eggs. Then virus isolation is performed in cell culture systems, and trypsin must be included in the culture medium, as most cell lines cannot cleave the hemagglutinin protein na nasa virion surface as discussed kanina, which is generally necessary for virus replication. The next, ginagamit din yung RT-PCR assays, lateral flow antigen capture test for laboratory diagnosis ng equine influenza viruses. The next, last, for the treatment, prevention, and control, halos naman sa mga viral infections, walang specific treatment. There is no treatment for equine influenza and more on supportive therapy lang siya. If may fever na more than 40 degrees Celsius na, usually NSAIDs ina-administer. If may nasal discharge naman or nag-lead na sa pneumonia, nagbibigay din ng antibiotics. Then, important din yung rest sa mga infected horses. Usually, at least 6 weeks na rest yung recommended for infected animals. This allows enough time para sa lungs nila na mag-recover sa infection. Then, available naman yung vaccination for prevention. Vaccination of equine influenza is common or extensively practiced in influenza virus and zoonotic countries, and inactivated vaccines are also used, pero may concern sa mga vaccine na to since they cannot provide a protective immune response for foals with maternally derived antibodies. To solve this, or as a recommendation, vaccines should be withheld until at least 6 months of age yung mga foals. The next, commercially available canarypox virus vector recombinant vaccines have been demonstrated to produce a cellular immune response in horses, and unlike inactivated vaccines, they can protect or it can be used in foals even in the presence of maternal antibody. Also, importante rin yung regular cleaning and disinfection ng mga equipments and transport vehicles sa farm for protection and prevention of infection, lalo na yung possible equine influenza at outbreaks na mangyari. So, ito po yung peak ng nasal discharge sa mga horses with flu, then lesions sa lungs. Then next, we have swine influenza virus. The first isolation of swine influenza was by Richard Shope in 1930. Other synonyms, we have hog flu, pig flu, swine flu. Then swine influenza viruses cause respiratory disease that can lead to outbreaks of influenza in pigs. And out of the 3 influenza types, we have A, B, and C. Only influenza A virus creates issues in swine, pero na-isolate pa rin sa mga pigs yung influenza B and C. Because of these, pigs have been considered the mixing vessel or for influenza virus because of their susceptibility to infection with both avian and human influenza viruses. Nakakapag-replicate rin yung influenza viruses of avian and human origin sa mga pigs. Then next, there are 3 common subtypes na nagkakos ng swine influenza. Namely, H1N1 subtype that can infect pigs and human also, but mild lang yung usually yung inaano nito. Then H1N2 that has been identified with both humans and pigs. And lastly, subtypes of H3N2 influenza A virus that can infect birds, humans, mammals, and causes severe flu seasons in humans. Then, for the distribution reservoir transmission, the virus was found to be regularly present in turbinates, tracheal exudate, and lungs of infected swine, but not in spleens, livers, kidneys, mucosa, brains, or blood. Kaya more on respiratory signs and symptoms minamanifest ng virus na to. Then pigs are also the most important reservoir for influenza A viruses since mas mataas nga yung susceptibility ng mga pigs sa mga human and avian influenza viruses. Then next, transmission, respiratory droplets or direct contact or pig-to-pig contact, and also indirect contact or mga fomites. The next, pathogenesis and pathology. Swine influenza viruses follows the typical pattern for respiratory viral infections since RESP virus enters via aerosols and meron ding rapid progression sa epithelium of the nasal cavity and airways. Incubation period is 24 to 72 hours with clinical signs of fever, apathy, hindi masigla, nangihina, inappetence, huddling with other pigs, and reluctance to move yung mga pigs. Meron ding signs of respiratory distress, paroxysmal or sudden attack of coughing and sneezing, rhinitis with nasal discharge, labored breathing, and bronchial rheals at auscultation. Some develop severe bronchopneumonia which may result in death. Yung disease ranges from subclinical to acute, though generally less than 1% lang yung case fatality rate, and other sources naman 1 to 4% lang yung mortality rate, but still there are reported cases na nag-result siya sa severe bronchopneumonia to death. There is also significant economic loss because of reduced weight gains of recovered animals, and pati na rin yung public health concerns. Dagdag ko rin, for the typical lesions of respiratory disease, makikita lesions in the apical and cardiac lobes of the lung with hyperemia and presence of inflammatory exudates sa airways. For the laboratory diagnosis, swine flu should be differentiated with infectious diseases caused by Actinobacillus pleuropneumoniae, Mycoplasma pneumoniae, mga respiratory related causing diseases na bacteria na na-discuss na natin sa bakte. Then virus isolation from nasal secretions and oral fluid using embryonated eggs or by cell culture. An RT-PCR test, serological test na hemagglutination inhibition, and lastly, ELISA that can be used to detect infections of unvaccinated swine. Then full genome sequencing and phylogenetic analysis can also be done Pero most common and rapid diagnostic method nga is RT-PCR. Then lastly, for my part, katulad ng equine influenza, there is no effective treatment. Although gumagamit din ng antimicrobials to reduce secondary bacterial infections, plus expectorants or mga cough medicine to relieve mucus sa mga airways may relieve severely affected herds. Then for preventive measure, may available na vaccination or vax. Sa mga sow, before farrowing, usually sila vinavax dapat para ma-maximize yung transfer of maternal immunity sa mga piglets. Gaya rin sa equine influenza kanina, sa mga foals, possible ang piglet but reduce efficacy due to issues sa maternal antibodies. Then, strict biosecurity measures and good management practices such as strict all-in, all-out procedures, limiting movement of pigs and sows, and preventing overcrowding sa mga pens.
Hi everyone, I am Melissa Mulle and I will be discussing avian influenza viruses. So first is avian influenza viruses, also known as bird flu. I think narinig nyo na din naman yung bird flu since dati nung may klase pa tayo sa poultry kay Doc Geng, na-discuss na din itong disease na to. So back in 1878, sa northern Italy, this disease was described as fall plague that causes contagious severe systemic disease with multiple organ failure and with high mortality rates. Before, nako-confuse pa yung mga tao with acute septicemic form of which is caused by a bacteria Pasteurella multocida. Pero kinalaunan, na-discover na this is actually caused by a virus, specifically type A influenza virus. So dati, ang tawag sa kanya is fall plague, but now it is called avian influenza since mas appropriate yung term. In third bullet, we have migratory birds na naka-emphasize. So ano nga ba yung role ng mga migratory birds na to? In 1961, nagkaroon ng outbreak of highly pathogenic avian influenza and low pathogenicity avian influenza virus sa may South Africa, na kung saan maraming namatay na common tern. Ang common tern is a strongly migratory seabird. Ito yung way kung bakit na-discover yung malaking role na ginagampanan ng mga migratory seabirds sa virus ecology, or basically kung paano kumakalat yung virus sa environment. Since yung mga birds na to, they undergo through seasonal migrations over large distances and nagkakaroon ng episodes of dead and dying migratory birds. This results in presence of the virus in new areas na kung saan nag-pose yun ng malaking risk or malaking threat sa mga commercial poultry industries. There are two clinical types of avian influenza that occur in poultry. The high pathogenic avian influenza that causes 100% mortality and morbidity, the low pathogenic avian influenza which causes asymptomatic infection. Since 1959 pa lang, nagkaroon na ng at least 35 outbreaks of highly pathogenic avian influenza virus infection. Lahat ng outbreaks na yon is nagmula sa mutant, meaning yung mga virus na to is kaya nilang mag-mutate. H5N1 A/Cus/Guangdong/1 and /1996. Parang complicated pakinggan, no? Pero paano nga ba nabuo yung name na yon? The letter A indicates that this is an influenza A virus. Gus naman is yung host species na kung saan first na-identify yung virus na to. And then Guangdong is the location or place kung saan first na-identify or na-isolate yung virus na to. And 1996 was the year naman kung kailan na-isolate itong virus. And this subtype again is the H5N1, which is the precursor and yung pinaka-nagtagal for 20 years na kumalat halos sa buong mundo na na-identify sa mga domestic waterfowl sa southern China, Guangdong. At first, hindi naman siya agad-agad na-detect but nag-emerge ulit siya sa China. Kumalat papuntang Asia hanggang sa Africa and papuntang Middle East and Europe noong 2003. And due to the gene swapping of H5 viruses, in 2014 to 2016, nagkaroon naman ng emergence of H5N6 and H5N8 HPAI viruses which became predominant. H5 viruses with various neuraminidase or NA genes continue to be detected, including in US wild birds and poultry. Ano naman yung role ng neuraminidase? So, bago makapag-infect ng other cells, di ba kailangan muna ma-release ng virus from the infected host cell? Bali, yung neuraminidase protein, inaalaw niya yung mga newly formed virus particles na mag-detach mula dun sa may host cell and mag-spread to infect other cells within the body. Mag-a-act siya as an enzyme and ito target niyang i-cleave or split yung sialic acid molecules na present dun sa may surface ng cell ng respiratory tract. While yung hemagglutinin naman is responsible sa pag-attach ng influenza virus sa host cell and siya yung mag-i-initiate ng process of viral entry. Magbabind siya sa may specific receptor na tinatawag nating sialic acid molecules na present dun sa may surface ng respiratory epithelial cells. Then, magkakaroon ng conformational change na mag-aalaw ng viral envelope na makapag-fuse dun sa may host cell membrane, releasing the viral genetic material into the cell. So basically, yung hemagglutinin yung nag-aalaw dun sa may virus na mag-stick sa may host cell and makapag-enter dun sa may host cell. While yung neuraminidase naman is yung makakapag-help sa virus na makalabas and makapagkalat ng sa iba pang cells. To sum it up, yung dating H5N1 virus lineage, nag-reassort na siya with different neuraminidase subtypes, giving rise to H5N2, H5N3, H5N5, H5N6, and H5N8 HPAI virus, and therefore it is herein designated as H5NX. And binabantayan na rin or minimonitor na rin yung mga trading between poultry and wild birds, whether legal man yan or illegal yung pag-trade, kasi yung H5NX infection na-detect na siya mula sa mga imported birds at international borders. Eventually, avian influenza virus led to millions of economic loss as well as millions of chickens and turkeys was killed due to the outbreak. Aside from that, the H9N2 and H7N1 subtypes are the low pathogenicity avian influenza virus. For the distribution, reservoir, and transmission naman, direct infection of individual humans by avian influenza viruses is sporadic. Although merong mga recorded na human infections and fatalities, very occasional instances lang, and it is mostly associated with H5N1 and H7N9 subtypes. So itong virus na to is nasashed siya in high concentrations sa mga feces ng mga wild birds and kaya niyang mag-survive for long periods sa mga cold water sources. Interspecies transmission can also occur, na kung saan yung pathogen nag-a-adapt siya sa new host species or nag-undergo siya ng genetic changes that enhances its ability to infect and spread. So from wild aquatic birds, especially sa mga wild ducks, to premises with mixed poultry species. And again, yung pinaka-common way for a virus to enter a territory is through migratory wild birds. Natatransmit din to through improper disposal of infected carcasses, manure, or poultry byproducts, pati na rin through fomites or yung mga insects and rodents, kaya nila lang i-carry yung virus na to. And ingestion or inhalation ng virus. And the H5N1 and H7N9 transmission to humans naman can be directly linked sa contact with infected poultry. Yung mga highly pathogenic avian influenza viruses can cause sudden death without prodromal symptoms. If the animal survive for 2 days after niya ma-expose sa virus, magkakaroon naman ng cessation of egg laying, respiratory distress, lacrimation, nagluluha, sinusitis, inflammation ng sinuses, and diarrhea, pati na rin edema or swelling ng head, face, and neck, and cyanosis of unfeathered skin, pati na rin dun sa may comb and bottle. If the animal survived 3 to 5 days after ng exposure, pwede siya magpakita ng mga nervous signs like tremors of the head and neck, hindi niya kayang tumayo, torticollis or yung neck twisting, and other unusual postures. Moving on to the pathogenesis and pathology, a key pathogenic marker of H5 and H7 viruses is yung amino acid sequence at the proteolytic cleavage site of hemagglutinin protein. Again, yung HA protein is a surface protein found on the influenza virus. Yung main job niya is tulungan yung virus na mag-attach and makapasok dun sa may host cell, particularly yung cells na naglaline sa may respiratory tract. Within the HA protein, merong specific spot or site na kung saan may specific amino acid sequence na kayang maklave or makat ng enzymes na tinatawag nating proteases na pinoproduce ng host organism. Ito yung tinutukoy natin na proteolytic cleavage site of hemagglutinin protein. It is important na maklave yung specific amino acid sequence para ma-activate yung fusion activity of the HA protein and ma-release yung viral genome into the host cell. However, if merong changes dun sa may amino acid sequence, pwedeng ma-alter yung rate of the cleavage of the protein and drastically alter the virulence of these viruses. Most LPAI viruses have single basic amino acid arginine at the cleavage site, and this can be cleaved by the extracellular proteases found in the respiratory or gastrointestinal tract of the birds. Yung ilang HA proteins, meron din silang glycosylation site that shields the cleavage site. Glycosylation site is where glycans attach and pinoprotect nila yung antigenic sites na ma-recognize ng immune system ng host, contributing to immune evasion and the generation of antigenic variants. While in HPAI viruses, the cleavage site contains multiple basic amino acids. Merong presence of several consecutive arginine or lysine residues. Dahil doon, yung HA protein can be cleaved by a wider range of host proteases, pati na rin yung intracellular proteases na present sa may variety of tissues and cell types. As a result, yung HPAI viruses, kaya nilang mas mag-replicate efficiently in multiple organs and tissues, leading to systemic infection and severe diseases in birds. Overall, yung hemagglutinin cleavage is a major determinant ng virulence of avian influenza viruses. Pero maliban pa ron, syempre meron ding ibang portions of the hemagglutinin protein bukod sa gene products na kayang mag-contribute through their respective roles on virus binding and replication efficiency. Meron ding variation when it comes to the susceptibility or resistance of different bird species to individual HPAI virus strains. Like for example, yung H5N1 virus na na-detect sa chicken in Scotland is more virulent for chickens, whereas yung H5N9 virus na na-detect sa turkey in Ontario is mas virulent para sa mga turkeys. If i-compare natin yung pathogenesis ng avian influenza, iba siya from that in mammals. Kasi yung virus replication ng avian influenza is sa intestinal tract pati na rin sa respiratory tract. In infections with most HPAI virus strains, there is viremia and systemic spread with hypercytokinemia or severe, which means merong severe immune reaction dun sa may katawan na kung saan nagre-release siya ng way too many cytokines, in which means merong severe immune reaction dun sa may katawan na kung saan nagre-release siya ng mas maraming cytokines dun sa may blood too quickly, proceeding with multifocal lymphoid necrosis and vasculitis and thrombosis and so on. For laboratory-based testing, gumagamit tayo ng real-time PCR assay to detect the M protein gene, as this is highly conserved in all avian and mammalian influenza viruses. If nag-positive yung sample, itatest ulit sila for specific H5 and H7 genes by RT-qPCR. If yung sample are H5 or H7 positive by RT-qPCR, sequence analysis is done to determine the properties of the cleavage site. Kapag naman nag-negative yung sample, pwedeng i-determine na lang yung HA subtype. If several basic amino acids are detected at the cleavage site, then the regulatory action is taken to eliminate the focus of infection. Nag-isolate din ng tracheal swabs and cloacal swabs to obtain viruses for antigenic analysis. This means na yung mga researchers, they determine how the virus is recognized and targeted by the immune system. And also for in vivo pathogenicity tests for researchers to understand kung paano nga ba or kung gaano nga ba ka-virulent yung viruses and kung paano ito nagkukos ng disease, which is essential sa pag-develop ng strategies para makontrol and makapag-treat ng mga infections. For the treatment, prevention, and control, yung treatment options for avian influenza virus infections, particularly sa mga birds, limited lang sila and usually nakafocus sa mga supportive care and management rather than the direct antiviral therapy. There are also public health concerns associated with HPAI viruses, as some strains have zoonotic potential and kayang mag-infect sa humans. And yung rapid progression and severity ng HPAI infections make it difficult to intervene effectively with antiviral treatments once yung birds ay infected na. Yung virus kasi is nagre-replicate rapidly and kayang mag-spread quickly within and between flocks. And some bird species may have receptors na mas compatible with certain HPAI virus strains kaya mas susceptible sila sa infection, while yung others naman is less susceptible kasi merong differences sa may receptor structure or distribution. Ito naman yung mga gross lesions of high pathogenicity avian influenza viruses H5N1 na naka-infect sa birds noong 2001 to 2002 season. Figure A, may makita tayong lesions na pancreatic necrosis, commonly multifocal to coalescing tan discoloration and occasionally hemorrhagic, yung tinuturo dun sa may arrowhead. Next naman is yung B, which is may makita tayong petechiae dun sa may chyliomic fat. Tingnan niyo yung arrowheads. And then, hemorrhage naman dun sa may proventricular mucosa centered on the proventricular ducts sa letter C na picture. And then, hemorrhages naman sa intestinal serosa dun sa may D na picture. And ito pa yung mga ibang informative pictures. Next naman is canine influenza virus or also known as dog flu. So, back in 2004, nagkaroon ng outbreak sa Florida kung saan yung mga dogs nagkasakit sila ng severe hemorrhagic pneumonia. And several dogs even died caused by a virus. And nung in-isolate yung virus na yon from an infected dog, na-discover nila na this is the subtype H3N8 virus na nagmula sa equine influenza virus type. And usually, yung tinamaan ng disease ng mga aso is yung mga nagkaroon ng close contact and yung mga inaalaga ang aso malapit dun sa area ng mga infected horses nung naganap yung outbreak ng equine influenza noong 2003. And all ages and breeds of dog are susceptible. Meron siyang two main subtypes. First is yung H3N8 canine influenza virus na kung saan itong virus na ito nag-originate sa horses and then nag-spread sa dogs. And now, kaya niya na mag-spread between the dogs. Since it is a highly contagious virus, usually yung canine influenza outbreak is nag-o-occur sa mga animal shelters, adoption groups, pet stores, boarding kennels, dog parks, veterinary clinics, kung saan merong close contact yung mga dogs. And the other subtype naman is the H3N2 canine influenza virus, which come from an influenza virus, H3N2, that adapted to infect dogs. And na-identify ito sa Korea. And kaya naman nitong mag-infect sa mga domestic cats. So for the distribution, reserva, and transmission, paano nga ba natatransmit yung canine influenza virus? Pwedeng through respiratory secretions like kapag kunwari umuubo yung aso or kapag nagsisneeze siya or even kahit tumatahol lang or kahit yung isang tao lumapit siya dun sa may or nakipagsalamuha siya dun sa may infected dog, yung ginamit niyang clothes pati yung hands niya pwedeng magkaroon na rin or ma-carry yung virus. Yung reserva ng virus na to are primarily dogs. Yung horses, sila yung original source ng H3N8 strain virus, while dun naman sa may H3N2 strain ay mga birds. Typically, inaabot ng 2 to 4 days after ma-expose dun sa may virus bago makita yung signs na infected yung dog with CIV, which is mild respiratory infection and pag-ubo na naglalast for 1 to 3 weeks. And yung way ng pag-ubo nila is same with the kennel cough. or canine infectious tracheobronchitis, which is typically harsh, dry cough that may be followed by gout. And usually, may kita mo naman yung mga signs or ways ng pag-spread ng virus. Pero there are also instances na kahit walang obvious signs, kaya pa rin kumalat ng virus. So for the pathogenesis, yung virus, mag-enter siya via inhalation ng mga respiratory droplets na nagko-contain ng virus. And then yung virus na yun, mag-a-attach siya dun sa may cells na naglaline sa may respiratory tract. And then, yung virus, mag-enter na siya dun sa may host cell and then dun na siya na magsa-start na mag-replicate. Yung virus na yun, i-infect niya na yung mga neighboring cells, saka siya mag-cause ng damage dun sa may respiratory epithelium. Clinical signs include respiratory illness, such as yung pag-cough na similar to kennel cough, sneezing, nasal discharge, fever, and lethargy. In severe cases, merong pneumonia. So for the laboratory diagnosis, virus isolation, polymerase chain reaction, serological testing, and immunofluorescence assay. Yung pag-treat sa kanila is more on supportive care. Like, kapag kunwari, syempre, weak yung immune system nila, syempre, ibuboost mo yon. Magpo-provide ka ng mga fluids, make sure mo na nakakapag-rest yung dog, and syempre, napoprovide mo din dapat yung nutritional care na kinakailangan nila, keep them comfortable. In severe cases involving pneumonia, pwede tayong mag-administer ng antibiotics. And for prevention and control, meron tayong mga available na vaccines for the canine influenza viruses. Siyempre, kinakailangan din na merong good hygiene, infection control practices, like kapag may sakit yung aso, ihawalay mo na siya dun sa mga healthy ones. And also, don't forget to disinfect the area. So for this slide, we have a comparison of gross pathological lesions ng lungs of the infected and uninfected dog. Yung left cranial, middle, and caudal lobes of infected lungs, meron tayong makikitang severe pneumonia along with edema and hemorrhage. Okay, next naman is bat influenza viruses. Unang na-discover yung bat influenza virus in little yellow-shouldered bats or Stornina lilium sa Guatemala during the time na nagko-conduct sila ng study, which is virological surveillance study, na ginagawa to track the presence, distribution, behavior, and evolution of viruses, including yung potential nila na mag-cause ng disease. So, na-detect nila yung virus na to, which is now called bat influenza virus, that has genetic and functional properties na same sa influenza viruses type A. And there are two known subtypes under the bat influenza virus. First is yung H1710 bat influenza virus. This is the one na na-detect from little yellow-shouldered bats sa Guatemala. And yung hemagglutinin and neuraminidase proteins nila were phylogenetically related to those of avian influenza viruses. However, yung twist lang is yung bat virus hemagglutinin does not mediate agglutination of red blood cells. And the neuraminidase does not sialic acid. So maybe meron silang ibang ginagamit na way to enter the cell and to leave the cell. And for the other subtype, which is H18N11 bat influenza virus, una itong na-detect sa mga rectal swab na kinolek from New World flat-faced fruit bats sa may Peru naman. And for the distribution, reserva, and transmission, yung distribution nila, generally, yung mga bats, they can carry viruses without showing symptoms of illness themselves. However, when virus jump from bats to other species, such as humans or other animals, they can sometimes cause disease outbreaks. But now, there is no known disease called bat influenza virus that affects human. Pero syempre kasi until now, nagko-conduct pa rin sila ng study or meron pa rin mga ongoing researches to understand the risk na associated with bat influenza virus transmission. And wala pa naman na-document na cases ng bat influenza virus na nagko-cause ng recognizable disease symptoms sa kanila. Transmission may occur through close contact, respiratory secretions, and possibly fecal matter. Close contacts, since yung mga colonies ng bat, tabi-tabi sila, so most likely nagkakahawaan talaga. For pathogenesis and pathogenicity, alam naman natin na yung mga bats. So for pathogenesis and pathogenicity, throughout sa evolution ng bats, nakapag-acquire na sila ng mga remarkable trait which is yung kanilang robust immune system, na kung saan inaalaw nun na makapag-coexist sila with a variety of viruses. Kabilang na ron yung bat influenza virus. That's why again, there's no showing of symptoms of illnesses themselves. Kasi kaya na nilang i-tolerate yung mga viral infections without mounting strong inflammatory response. Basically, they don't respond to infections the way we do. Although yung bat influenza virus have not been known to cause widespread illness in humans, still, they pose a zoonotic risk. They have the potential to infect and cause disease in humans, since yung influenza viruses nga have the potential to undergo genetic changes that could allow them to infect and spread among humans. And with that, further research is needed. For laboratory diagnosis, we conduct virus isolation, RT-PCR, qRT-PCR, ELISA, and hemagglutination inhibition ASA. There is no specific treatment or vaccines na na-approve for BIV, and for prevention and control, syempre importante yung surveillance and monitoring to detect any emergence or changes dun sa may viral activity. Moving on, we have bovine influenza V virus. Merong newly recovered virus sa may respiratory tract ng cattle sa United States na napansin nilang may pagkakapareho or similarities sa human influenza type C virus. Pero, upon further checking, they eventually realized based on the genes and kung paano siya mag-behave and napagdesisyonan ng International Committee on Taxonomy of Viruses na it was different enough to create a new group kaya tinawag siyang influenza virus D. So bale, yung new group is named after the type species, which is the bovine. Kaya bovine influenza V virus. And usually, nadidetect tong virus na to sa cattle with respiratory disease. So for distribution, reserva, and transmission, yung infected cattle kaya niyang i-harbor yung virus and i-shed yun sa environment. It can also infect small ruminants like sheep and goats. Virus infects swine, albeit with much lower frequency than cattle. So, kaya niyang i-infect yung swine, pero hindi kasing lala nung sa may pag-infect niya sa cattle. And bovine influenza D virus are not known to infect humans. Pero yung transmission to other animals may occur through close contact, inhalation, and fomites. Yung pathogenesis and pathology naman, mag-enter yung virus through inhalation, and then mag-a-attach dun sa may epithelial cells na naglaline sa respiratory tract, and then mag-replicate na siya, and then magre-result yun sa pagka-damage ng mga infected cells and the release of viral particles into the respiratory secretions. So yung virus kaya niya na mag-spread locally within the respiratory tract and systemically via the bloodstream, which can now lead to the infection of other organs pati ng mga iba pang tissues. So for the clinical signs, respiratory illness, kaya ng fever, coughing, nasal discharge, and hirap sa paghinga. And in severe cases, merong pneumonia and secondary bacterial infections may develop. So for laboratory diagnosis, we conduct molecular tests including yung RT-PCR for viral detection and serological assays for antibody detection. So for the treatment, more on supportive therapy or supportive care para maibsan yung nararamdaman and mabilis na makapag-recover yung cattle. Aside from that, for prevention and control, vaccines are available. And of course, kailangan mahigpit yung biosecurity measures and surveillance and monitoring of influenza virus activity sa may cattle populations. And last, for my part, is the human influenza virus. Influenza virus A, B, and C are known to infect humans. Like yun sa may influenza virus na binanggit ni Jaisal kanina, which is yung classifications nila. Influenza virus A commonly infects humans, horses, swine, and domestic poultry. While sa may influenza virus B naman, it commonly infects humans. And influenza virus C commonly infects humans and swine. But generally, epidemic human influenza viruses typically do not persist or circulate wildly among animal populations. Pero syempre, meron pa rin mga exceptions ron. Like, yung sa pigs, the H1N1 under ng swine influenza virus na nag-emerge noong 2009 that caused influenza pandemic, and it was believed na na-transmit lang talaga sa pig yung virus. Since yung pig, they are susceptible to human and avian influenza strains na kung saan yung mga pigs nagsiserve sila as mixing vessels ng virus. Gaya nga ng binanggit ni Jazel, yung mga ferrets din is pwede din mahawa and makapag-transmit ng both human influenza A and B viruses. Ginagamit din sila bilang laboratory models para sa mga pathogenesis studies because the associated disease closely mimics that in humans. Non-human primates, including gibbons, baboons, chimpanzees, can also be naturally infected with human influenza A virus. So for the distribution, reservoir, and transmission, ang primary reservoir is us, humans, and then it can spread from person to person, and kaya niya rin mag-infect ng animals. Like kanina, di ba yung binanggit sa pigs, and syempre close contact with infected individuals or sa mga contaminated surfaces, or kapag nakapag-inhale ka ng mga respiratory droplets. For pathogenesis and pathology, of course, via inhalation ulit and then attachment to sa may epithelial cells ng respiratory tract, and then it will enter the host cell, it will replicate and produce more viral particles. And then the infected cells will eventually burst, kaya magre-release siya ng bagong virus particles na kayang mag-infect sa mga neighboring cells. So for the symptoms, headache, lagnat, tiredness, pananakit ng mga kasukasuan, nasopharynx, runny nose, stuffy nose, sore throat, coughing, susuka. So for laboratory diagnosis, we conduct PCR and ELISA. Treatment includes antiviral drugs, Tamiflu, Relenza, Rapivab. And syempre, kapag may lagnat, para mabumaba yung temperature, gagamit tayo ng acetaminophen or ibuprofen. And syempre, para ma-relieve din yung pain and muscle aches. Syempre, kailangan din ng adequate hydration and rest at home. Syempre, kapag may sakit ka, 'di ba, as much as possible, magstay at home ka na lang para ma-lessen yung possibility na makahawa ka sa iba. And that's all for my report. Thank you so much for listening.
So now let us continue to the family Pneumoviridae. I'm Artreth Miklat from DPM3B. So first, let us talk about the taxonomy of this family. So this taxon was formerly a subfamily within the Orthomyxoviridae but was reclassified in 2016 as a family with two genera. These are Orthopneumovirus and Metapneumovirus. So first, the members of the genus Orthopneumovirus infect only mammals, while the members of the genus Metapneumovirus are specific for mammals and birds. So some viruses are specific and pathogenic for humans, such as human respiratory syncytial virus and human metapneumovirus. There are no known vectors for pneumoviruses, and transmission is thought to be primarily by aerosol droplets and contact. So let's move on to the taxonomy. First is the realm Riboviria, kingdom Orthornaviridae, phylum Negarnaviricota, subphylum Hapleviricotina, class Mongiviricites, order Mononegavirales, and family Pneumaviridae, and these are the genera which are the Orthopneumovirus and Metapneumovirus. For its for its species we have for its species we have three for genus Orthopneumovirus and two for Metapneumovirus. So the first for Orthopneumovirus is the Orthopneumovirus bovis, which is named bovine respiratory syncytial virus, and the second one is the Orthopneumovirus hominis, which is named human respiratory syncytial virus, and the third one is the Orthopneumovirus muris, which is named murine pneumonia virus. And let's proceed under the genus Metapneumovirus. First is the Metapneumovirus avis. which is named avian metapneumovirus, and the second one is the metapneumovirus homies, which is named human metapneumovirus. And these viruses will going to be tackled later on. So we also have unclassified species, which are the caprine pneumovirus or the CRSV and the ovine pneumovirus, ORSV, and the canine pneumovirus, CPV, which I will going to also talk about later on. So let's proceed to our first species. So bovine respiratory syncytial virus. It is under the genus Orthomyxoviridae, and it's also called Orthomyxovirus bovis, or its binomial nomenclature. So it is now known to occur worldwide in all bovine species, but it is not limited to that because it also can infect sheep, goats, and other ungulates. Some antibodies developed to detect the human respiratory syncytial virus can cross-react with the bovine equivalent due to their close relation. So, caprine and ovine strains of respiratory syncytial virus may represent, along with bovine respiratory syncytial virus, a subgroup of ruminant syncytial viruses rather than different species. So, for its clinical features, it causes pneumonia, pulmonary edema, and emphysema. The infection is characterized by a sudden onset of high fever, hyperpneumonia, abdominal bleeding, lethargy, rhinitis, nasal discharge, and cough. The virus persists in herds, most probably through continuous subclinical reinfections or in putatively inapparent virus carriers. So most affected are those recently weaned beef, calves, and young cattle. So take note of that. The most affected ones are the recently weaned beef calves and young cattle, especially when they are maintained in a confined environment. So secondary bacterial pneumonia that is caused by Mannheimia hemolytica is also common and high morbidity and low mortality. So those animals that died are known to have infected with bovine viral diarrhea virus. So the lesions are destroyed cilia within the airway epithelium. When cilia are destroyed, the pulmonary clearance is compromised, which predisposes to secondary bacterial infections. And interstitial pneumonia with emphysema that affects all lobes of the lungs. And another one is the syncytia, which is the characteristic of these viruses. Syncytia may be present in the airway epithelium lining, the bronchi and bronchioles, as well as in alveolar macrophages and type 2 pneumocytes. So these are the lesions that was mentioned. These are the pictures of these lesions that are mentioned a while ago. So we have here the emphysema of all the lobes of the lungs and interstitial pneumonia. And also here we also can see the syncytia, which is the characteristic of this virus. So let's move on to the diagnosis. Isolated from appropriate samples using several bovine cell cultures, which because we are growing a virus here, we cannot use media. So agar media, for example. So we can also diagnose from lesions and immunofluorescence staining of tracheal wash-derived cells, virus-specific monoclonal antibodies, and in tissue samples from necropsy cases. We can also use RT-PCR test. And virus neutralization assays can also be used to detect neutralizing antibodies. So the transmission, it spreads rapidly through aerosols or droplets of respiratory tract excretions. That's why outbreaks occur in herds kept in a closed environment. So the treatment, there is no specific treatment for the viral interstitial pneumonia, but suppurative treatment and correction of dehydration may be necessary. And gamithromycin is used to treat the secondary infections. Take note that that we can just use antimicrobials, antibiotics when there are presence of bacteria and not viruses, such as Mannheimia hemolytica, Pasteurella multacida, and Histophilus somni. For the prevention and control, the pre-existing antibodies, whether derived passively from maternal transfer or actively by prior infection or vaccination, do not prevent virus replication and excretion, although we can reduce the severity of the disease. So, several inactivated and live attenuated vaccines are currently in use. So next species or Next species, we have here the human respiratory syncytial virus, which is also under the genera of Orthomyxoviridae. So, Orthomyxoviridae hominis is also the binomial nomenclature of this virus. So, major major cause of acute lower respiratory tract infections worldwide in infants and children younger than 2 years old. We have to take note of the age of the affected individuals here, and major public health problem and economic burden in most countries. So elderly and immunocompromised individuals are also affected. So clinical signs, it is characterized by an exacerbated pro-inflammatory and unbalanced Th2 type immune response, and adverse effect in airway tissues like rhinorrhea, cough, congestion, low-grade fever, and respiratory distress, to severe alveolitis, bronchiolitis, and pneumonia. We can also take note of the neurologic manifestations such as seizures and encephalopathy. So for the human respiratory syncytial virus, we have here the risk factors, just like preterm birth, congenital heart disease, chronic lung diseases, and immunosuppression. For its lesion, we have bronchiolitis. It is referred to the distal bronchial inflammation and obstruction. For its transmission, it spreads rapidly and efficiently throughout the population by inhalation of aerosolized droplets of infectious viral particles, and also direct contact of these droplets with the ocular mucosa. So for the diagnosis, we can use blood tests to check white cell counts or to look for viruses, bacteria, and other germs. And chest x-rays to check for lung inflammation, and also swab for secretions from inside the mouth or nose to check for signs of the virus. So for the treatment, there is no specific treatment here, and but we can use intravenous fluids to aid in hydration, and breathing machine or humidified oxygen to help the body receive the oxygen it needs. So prevention and control, as humans, we can perform hand washing and avoid touching our face and avoid close contact with others, and we have to be vaccinated also, and monoclonal antibody products are available to protect infants and young children from severe respiratory syncytial virus. Now let us proceed to the murine pneumonia virus. It is under the genus of Orthopneumovirus, and it is also named as Orthopneumovirus muris. It causes murine pneumonia, and aside from mice, it also infects rats, cotton rats, genus Sigmondia, hamsters, subfamily Cricketinae, gerbils, subfamily Gerbillinae, guinea pigs, Cavia porcellus, and dogs. It is known to be non-cytolytic, and it infects respiratory epithelium and type 2 pneumocytes. So for its clinical signs, pneumonia in immunodeficient mice, and may exacerbate pneumonia caused by either pneumocystis pneumocystis species, or bacterial infections. Immunodeficient mice can have a progressive interstitial pneumonia with wasting. So, for its lesion, there is no recognizable necrotizing lesion because they infect the individual cells itself. So, for the transmission, the aerosol and the direct contact with respiratory secretions are the predominantly mode of transmission. So for the diagnosis, PCRs for the detection of active infections, and it is for testing trachea or lungs, and in the sites of infection serve as optimal samples. So also we can use ELISA for the differential diagnosis. We have here the Sendai virus, the pneumonia virus of mice. So it infects entire respiratory epithelial population and has recognizable necrotizing lesions unlike the murine pneumonia virus. So for the treatment, we have intratracheal hinokitol. Hinokitol is the treatment that lowers the levels of inflammatory cytokines in BALF and serum. So, excessive production of inflammatory cytokines or chemokines is a hallmark of tissue injury and severe pneumonia. So that's why we are administering intratracheal hinokitol. So for the prevention and control, we have intranasal murine pneumonia virus vectored SARS-CoV-2. So additionally, we have here the histologic view of the affected pneumocytes of the lungs. So next, we have the canine pneumovirus. So unlike the other viruses that were mentioned before, they were all under the genus Orthomyxoviridae. But now let's proceed to the unclassified genus, and this species is called canine pneumovirus rather. So it causes canine pneumonia and it contributes to the canine infectious respiratory disease or CIRD complex. It is a group of viruses and bacteria that comprise the multifactorial etiology of canine upper respiratory diseases. So, canine pneumovirus is difficult to isolate in cell cultures, and symptoms are fever, coughing, sneezing, and nasal discharge. It is transmitted through aerosols and direct contact from discharge and PCR tests to diagnose, which we can treat by doxycycline or amoxicillin clavulanic acid for 5 to 7 days from secondary infections or from the bacterial infections that comprises this complex or the seed. So we also have here the avian metapneumovirus under the genus Metapneumovirus, and it is known as Metapneumovirus avis. It causes avian rhinotracheitis and has types A, B, C, and D. So in the first picture, we can see the turkey here, and the second picture, we have here the chicken. So later on, we're going to talk about the diseases caused by this metapneumovirus, avian metapneumovirus. So here's a table that presents the different types of avian metapneumovirus and its corresponding disease with their animals affected and the area it was observed. So first we have the swollen head syndrome in chickens. Pardon for the wrong spelling here. We're going to correct it later. So it is caused by type A metapneumovirus. And next is the turkey rhinotracheitis, which has a other name of avian pneumovirus infection of turkeys. It is caused by type A metapneumovirus also. So let's proceed to the clinical features of these diseases. For the turkeys, young turkeys, The disease is characterized by inflammation of the respiratory tract, rales, sneezing, frothy nasal discharge, conjunctivitis, swelling of the infraorbital sinuses, and submandibular edema. For the three turkey breeding operations, there is a decrease in egg production of up to 70%, which is high. So there is also an increased incidence of the prolapse uterus due to excessive coughing. So for the tissue of the turkey animals, the respiratory tract disease is characterized by it is characterized histologically by increased glandular secretion, focal loss of cilia, hyperemia, and mild mononuclear mucosal inflammation within the turbinate during the first 2 days after infection. Also, epithelial destruction and intense mucosal inflammation on days 35 and watery to mucoid exudate in the turbinates from days 1 to 9. So, tracheal lesions are generally milder, but in severe cases can include complete dissiliation of the mucosal lining of the trachea within 4 days. So, cytoplasmic eosinophilic inclusion occur in epithelial cells lining the airways and nasal cavities. So, for the clinical feature of chicken, we have older pulse, which is characterized by coughing and head shaking, are frequently observed, may exacerbate by secondary infections. And for the hens, respiratory disease is milder than in young poults. Morbidity is often 100%. Mortality ranges from 0.4% to 50% and is highest in young poults. So, swollen head syndrome is a milder form of the disease, typically with co-infection by bacteria such as Escherichia coli. The disease is characterized by swelling of the infraorbital sinuses, torticollis, disorientation, and general depression, sometimes also with respiratory distress, and in chickens, morbidity is usually less than 4% and mortality is less than 2%. So, diagnosis for both turkeys and chickens, we can use ELISA and RT-PCR in acute respiratory disease cases and isolation by serial passage in 6 to 7 day old turkey or chicken embryos or in chicken embryo tracheal organ cultures. For prevention and control, we have live attenuated and inactivated vaccines and we have them commercially available for three genetic subgroup types of avian metapneumovirus A, B, and C. So it provides cross protection against the virus strains. So next we may proceed to the human metapneumovirus, which is under the genus of metapneumovirus and is also named metapneumovirus hominis. It causes human metapneumovirus infection and it can cause similar symptoms as respiratory syncytial virus, but the peak age for the severe illness for human metapneumovirus infection is between 6 and 12 months, but the respiratory syncytial virus is more likely to cause severe illness in infants younger than 6 months. That's why it is important to differentiate these two viruses. So symptoms, we have cough, fever, runny or stuffy nose, sore throat, wheezing, shortness of breath or dyspnea, and rash. For the transmission, coughing and sneezing, shaking hands, hugging or kissing on touching surfaces or objects like phones, door handles, keyboards, or toys. For the risk factors, those are younger than 5-year old or older than 65 years old. So have a weakened immune system from conditions like HIV, cancer, or autoimmune disorders, or from medications that suppress your immune system. Also have has asthma or COPD. So further complications which can occur are the bronchiolitis, bronchitis, pneumonia, asthma or COPD flare-ups, and ear infections or otitis media. For the diagnosis, based on symptoms and history, swabs for laboratory tests, and we can also perform bronchoscopy or chest x-rays to look for changes in the airways of your lungs. So treatment, oxygen therapy, IV fluids, and corticosteroids. So prevention, washing our hands, covering our nose and mouth, avoid being around other people when they are sick with cold, and considering wearing a mask and avoid touching the face and don't share food or eating utensils with others. So we have here the summary of the discussion I've tackled before. So first we have the genus Orthomyxoviridae. So we've tackled the bovine respiratory syncytial virus, which is also called Orthomyxovirus bovis, and its name it causes bovine respiratory syncytial virus infection. Also, we have talked about the human respiratory syncytial virus, which is also called Orthopneumovirus hominis, which causes acute lower respiratory tract infections. And we also talk about murine pneumonia virus, which is named Orthopneumovirus muris, and it causes pneumonia virus of mice. It causes pneumonia of mice. We also have here the genus Metapneumovirus, which we have talked about the avian metapneumovirus, which is also named Metapneumovirus avis, and it causes avian rhinotracheitis, and human metapneumovirus, which is also named Metapneumovirus hominis, which causes human metapneumovirus infection. So that's all for our report. So for the Orthomyxoviridae and Pneumoviridae. Thank you very much.